The diagram shows one way that stem cells can be produced from human embryo. You have to fertilize the egg, and when the embryo is formed after 5 days, you get the inner cells, and then you get the stem cells and all of that. What are stem cells? What are stem cells?
Sir,
Yes.
Aren't they undifferentiated cells that have the potential to like develop into any other type of cell?
Cells. Cells. That's it. Stem cells are undifferentiated cells
Oh, wait. That have the potential to develop into many different cell types.
That is your definition of the stem cell. Undifferentiated or pluripotent. Pluri means many. Pluripotent means many potential. Many potential to do what? To become any type of cell or any type of tissue or organ, or even depending on the level at which so the stem cell was harvested, can develop even into a whole organism. Now, where does this actually come from? You see, when there is fertilization, an ovum fertilized by a stem cell, it produces a zygote. The zygote first is just one cell, but then that zygote grows, divides the first time to form 2 cells, divides the second time to form 4 cells, that's the 4-cell stage. If I leave it at this point here, let's say this was happening in a human, we know clearly that this group of cells one day will become a fully grown person or a fully grown baby. But it started, let's say, as 4. So we ask ourselves, with these 4 cells, do we know exactly, if I call them, let's say, cell A, cell B, C, and D. Do we know which cell will produce the head of this individual between A, B, and C? Anyone? Do we know which cell between A, B, C, and D will produce the head of that person?
No,
Yes,
sir.
no? Yes, no?
No.
No. Do we know which cell will produce the tongue?
Yes.
No. The liver? No. The skin? No. But all we know is that these 4 cells, if they are left to continue to develop normally, they will grow to become a full individual. But again, we confirm that we cannot tell exactly which cell will do what. Now, the fact that we don't know which cell will do what, yet that we are sure that they can produce a single person, it means that these cells, all of them at this point, have the potential to produce anything in that human being. In fact, to produce even a whole human being on its own. If I was to separate these 4 cells, split them, cell A, if I put it alone, it will still grow to form one person. Cell B, if I separate it, it will still grow to form another person. Cell C, a different person. D, another person. But if you leave them as they were together in one group like this, they'll form only one person. If you split them at this age, they'll form 4 different people instead of just one. Why is that so? The reason is because they still have all the genetic instructions, very unspecialized instructions that still can produce anything, cells, tissues, organs, systems, organisms. That's what we call them stem cells. That's what we refer to as undifferentiated. When we say undifferentiated, for example, it means what? This group of cells here cannot be any particular cell that we can think of. Different cells that we have in our body, we have maybe ciliated columnar epithelial cells, we have cells of the nervous tissue, like the neurons. We have, for example, cells like even stem cells, ova. These are all different cells. But these cells that we see here are none of those yet. It means they have not yet differentiated. They've not yet specialized to become maybe a stem cell, to become maybe a nervous tissue cell, to become maybe a muscle tissue cell or a skin cell or an epithelial cell. They've not yet specialized. But when they specialize, it means they differentiate. But once a cell has decided to become a liver cell, it can no longer become a skin cell. It has already chosen that path. Once it has decided to become the cell of the pancreas, it can no longer become the cell of the tongue. It has chosen already a path. That's what we mean by differentiation and specialization. So our stem cells here have not yet reached that level. It means also that we can use them and do anything we want to do with them. Just like a child who is still at creche, that child has the potential to become anything in life. The child can become a soccer player, a netball player, a basketball player, a swimmer, a lawyer, a pilot, a scientist, medical doctor, anything. But the moment that child starts growing, reaching around stage grade 10, the child starts specializing, either commercial or science stream. Going again, the child has already reached maybe second year university and has never played soccer in his or her life. That child can no longer come back and say, you know what, I want to have a professional soccer career. Not really possible anymore. So you see, the older we get, the smaller our career options become. The same thing with stem cells. The older they get, from embryonic stem cells, like you see here, from embryonic stem cells, you'll find some of them also in the blastocyst. The older they get, the more they lose that potential to become anything again. Because you can harvest stem cells from 3 major sources. Either you harvest them from the embryo, that is the youngest one. It's just like you're taking a child first year at fresh. Very, very fresh can become anything. That's the embryo. This is maybe grade R, embryonic stem cell. Now, when a child reaches grade 8, can still become anything, but cannot become anything like a child in grade RRR. This one now will be stem cells that we get from maybe cord blood. Cord blood is the blood that is squeezed out from the umbilical cord after a child is born. And maybe you get now a child at grade 12, career options again as reduced. This is the kind of stem cells that we get from bone marrow. So the stem cells are of different grades. The best grade of stem cell, therefore, is the embryonic stem cell. That is the best grade because you can use it to produce virtually anything. So that was just background information about what stem cells are like. You can look at the question now. Now, to define a stem cell, give 2 sources of stem cells other than human embryos. I've actually answered it. Can we remember 2 sources? 2 sources of stem cells other than embryonic. Please, let's be fast. Hello? Are you guys listening or talking to myself?
You're listening, sir.
Yes. So sources of stem cells other than the embryo?
Bone marrow,
Thank you. Bone marrow. What else? Thought, thought what?
blood,
Blood, blood. Blood, and bone marrow. What are the other people? Pick one medical condition that can be treated by using nerve cells grown from stem cells. Actually, I need two. That is done. Medical conditions that you can use nerve cells to treat are multiple what?
so is it multiple sclerosis?
Of course, multiple sclerosis and Alzheimer's disease. If you remember your multiple sclerosis and Alzheimer's disease, it is said there is no treatment, but stem cell research holds a promise for this. The reason is because multiple sclerosis is caused by damage done to myelin sheath of neurons in conductive conduction pathways. Meanwhile, Alzheimer's disease is also caused by loss of either neurons or loss of cell bodies or entangled neurons. Either ways, neurons are damaged. What can I do with stem cells? I have heard that stem cells can become anything. I get a stem cell, I give it direction, I groom it to become a nerve cell that is still functioning. Then I just go and replace it. I go and I replace it. This is what I'll do, pure picture of the person. That is what is still being underway. I don't know if they consider to do any of such, but since they know the potential of stem cells, they can use probably such thing to that. Okay. Human blood type is an example of multiple alleles. Why are blood types examples? Today, what I'm doing today, I'm not doing topic per topic. No. I've taken maybe one or two question papers that were already written, past papers. Then we're going right through the paper so that I can easily explore every topic at the same time in one paper. So, why are blood types an example of multiple alleles? I'm waiting. Why are blood types an example of multiple alleles? Please, what I will type here, I need you guys to take a screenshot after I'm done typing. Okay, you can all hear me now, right? I was saying, I was busy typing this so that you guys can take a screenshot. I'll explain now. There are, according to the ABO blood group system of classification in humans, there exists 4 blood groups, A, AB, and O. The blood groups are controlled by 3 alleles. It is I, A, I, B, and small I. Each individual can only inherit 2 of the alleles. That's why you can have someone who is either this, IAA, for example. The person has only 2 of the alleles, or the person can be IA, IB, still only 2 out of 3. An individual can only belong to one blood group. That's why a person can only be either blood group A, or blood group B, or blood group AB, or O. So if you observe what we've done here, we've done a countdown from 4, 3, 2, 1, so that it will be easy for you to remember. Now, the question said, why are blood groups considered as or as having multiple alleles? If you check the normal Mendelian crosses, if we were talking about height, it was always either tall or short. It was just 2. But with blood groups, we've seen how many? We've seen 3. 1, 2, and 3. 3 is more than 2. It is referred to as multiple. That's what it means. Is it clear to us? No? Did you guys hear me?
Yes, sir.
Yes, sir.
Please, can you guys just say yes when I'm asking? It's like only one person is hearing me speak. I need to be sure that I'm not mute or something.
Yes, sir. I'm not sure I could hear you.
Okay. So, give the possible genotypes of a man who is blood type B. Then, another table. Yeah. Our possible genotypes for all of them. Whether it's blood type A or what. Genotype or blood group. Blood group is a phenotype. Here we have possible phenotype or phenotypes. So, it is blood group A, B, AB, and O. The possible phenotypes here. For A, it will be I, capital A, or capital I, capital A, and small I. And B, capital I, capital B, capital I, superscript, capital B, or capital I, capital B, lowercase I. AB would simply be capital I, superscript A, capital I, superscript B. And O is double recessive B. So you'll realize that for blood group A, it can be homozygous or heterozygous. Blood group B, similarly, homozygous or heterozygous. That's how the blood groups are. So, if a man of blood has blood group, if a man has blood group B and a woman has blood group A, explain how it is possible to have a child who has blood group O. Do not draw a genetic diagram. How is it possible? How is it possible to have a child with blood group O from a man of blood group? Yeah? A man who has blood group B and a woman of blood group A. Explain how it is possible to have a child who has blood group O. That's our question. So, they said do not draw a genetic diagram. It's out of 3 marks. Nevertheless, the man is blood group A, the woman is blood group B. We see that for each of these blood groups, there are 2 possibilities. It's either we use this and cross with this. Of course, it will never give us a blood group O because blood group O needs this kind of genotype. So if we cross these 2 here, we cannot produce this. So although they are blood group A and B, we shall only use the heterozygous blood groups. That's what we do. So you'll say the parent must be heterozygous for blood group A and B. Or you say the parents must be the parents must be I or now. The parents must be I, A, I, or I, E, I. You can say that. And you say the child will receive a recessive allele from each parent. Or you say the child will receive I from one parent and another I from the other parent. These are already 2 marks here out of 3. 2 marks. The first part here where you talked about the genotypes of the parent heterozygote or IAI, Then the second part where you talk about what the child receives. The third mark is when you say the genotype of the child will be II. That's the third mark. And we just conclude by saying the child will be of blood group O. That's the 3 mark. Can take a screenshot, please. Okay, let's try and do also one investigation question. Not on any. But we see how we look for what we are being asked. Many athletes have to train hard to build muscles. Muscle is a protein and is therefore formed by protein synthesis. Athletes need to eat and train to ensure optimal muscle protein synthesis. They always want to know how much protein they must eat to build bigger muscles. Scientists carried out an investigation to determine the optimal daily protein in grams needed for maximal increase in muscle protein synthesis, that's MPS. The investigation was carried out as follows. They studied 100 bodybuilders. The moment I see 100 bodybuilders, it tells me that this is what? This is a large sample size. And then it tells me that this helps increase reliability. Reliability is increased by either having a large sample size or repeating the investigation. Each bodybuilder was given the same daily weight lifting exercise. The moment I see same, it tells me that this is a controlled variable, and it tells me that we are trying to make the experiment complete. Each time it increases validity. So same is for validity. Same is for controlled variables. And it is controlled variables when they are kept constant that increase validity. All athletes were fed the measured protein meal straight after exercising. The MPS was recorded 4 hours after exercising. The graph below shows the results of the investigation. These results were similar to other 2 investigations carried out by other scientists. And I need you to note this part here. Similar to other 2 investigations. Just note it, we shall come back to it. So we have a graph here. First question. Identify the conclusion reached by the scientists was as follows. The optimal daily protein needed for maximal increase in muscle protein synthesis is 20 grams. Set the dependent variable and independent variable. So look at this and tell me which one is the dependent, which is the independent. Which one is the dependent, which one is the independent? Can someone tell me what is the aim of this? What is the aim of this investigation? What's the aim of this investigation?
So,
Yes,
um, can I try?
yes, please.
So isn't the dependent variable like how much protein they need to eat in order to build bigger muscle?
Not exactly. Can you start by telling me the aim of this investigation?
So the aim is that they want to know how much protein they must eat to build bigger muscle.
The aim is to determine the optimal daily protein. Yes, I'm glad when you say how much protein, but then I need you to say the amount of protein. The aim is to determine
Okay.
the optimal daily protein needed for maximal increase in muscle protein synthesis. Okay, so why I'm
Yes.
asking you guys to identify the aim is so that you guys must know whenever you are asked for the dependent or independent variable, please, the first thing you must look for is the aim. Once we've identified the aim, from there, I can pick out my dependent and independent variables. We are going to be kicked out very soon. When we get kicked out, please log in again in 10 minutes for the last session. So this is my aim here to determine this. Therefore, my dependent variable will be muscle protein synthesis is dependent. The dependent variable is muscle protein synthesis, that's the dependent. And my independent variable is amount of protein. That's optimal daily protein. Amount of protein is independent. Muscle protein synthesis is the dependent because muscle protein synthesis depends on how much protein these people are eating. Is it clear? Okay, log in 10 minutes, please.